Hi everyone, this is Samandeshwari and here is my partner Sugita. This is our final year project presentation which was carried out in Central Leather Research Institute, Chennai. Our external guide is Dr. D. Murugan, who is a scientist in CLRI, and our internal guide is Mr. A. Meenakshi Sundar, who is an assistant professor in Vailtech High Tech Engineering College. Our project title is Effect of Surfactants on Anaerobic Co-Digestion of Sludge from Tannery Effluent Treatment Plant. Let's have the brief introduction about our project. Energy crisis and waste management are the two important problems faced by the world today. Excess sludge, which is an unavoidable byproduct generated in common effluent treatment plants involved in the treatment of tannery wastewater. Excess sludge treatment and disposal requires about 60% of total plant operating cost. So there is a worldwide search for the solution which is both energy efficient and cost effective. Anaerobic digestion proves to be the most promising technique to the disposal of these kinds of waste because of its high energy recovery. Anaerobic co-digestion is nothing but the degradation and destruction of organic substances with consequent reduction of odor and pathogens in the absence of oxygen. In the literature survey, we found that addition of surfactants in lower concentration increases the biogas production during the anaerobic digestion process. Objectives of our project are to identify the effect of surfactants on the activity of microorganisms using isothermal microcalorimeter, to verify the process concept in terms of gas production in the lab scale digester, to study the effect of surfactants, that is both natural and synthetic surfactants, and their influence on biomass activity and gas production. Here are some of the papers which we reviewed for our project. The first paper examines that the addition of LAS to the anaerobic digester results in the increase of biogas production. The next paper studies the effect of alcohol sulfate, which is an anionic surfactant, and the effect of biodegradable compounds on the anaerobic digestion process. The next paper demonstrates a gradual conversion of organic substances by methogens during the digestion process. Methogens is nothing but the methane producing organisms. The next paper provides a complete kinetics and metabolic analysis involved in the anaerobic co-digestion process. The next paper deals about the removal of anionic surfactants present in the activated sludge system. And the next paper gives us the complete review on anaerobic digestion and anaerobic co-digestion process. These are the materials which we used for our project. Secondary sludge obtained from the tannery effluent treatment plants is used as substrate. Cow dung, which is inoculated for more than 15 days, were used as a biomass. 3 surfactants are used, that is saponin, sodium lauryl sulfate, and Tridonex 114. Saponin is a natural non-ionic surfactant which was extracted from soap nets, whereas SLS and Tridonex is a synthetic surfactants and are obtained from Sigma Aldrich, Mumbai. Next we will see the characteristics of sludge and biomass. The total and volatile solids for the biomass and secondary sludge and the mixture of both were determined using the general methods and their values were shown in the slide. Let's move on to the methods. Saponin was extracted from Sapindus macrosy, that is nothing but soap nets, using the methods described in the literature. The procedure is that the saponins were de-seeded, dried under sunlight for more than 4 days. The dried pericap is leached with water and then centrifuged at 3000 rpm to remove insoluble matters. Then the extract is added with ammonium sulfate to precipitate the saponin. The precipitated saponin is then filtered off and then washed with water. Then it is dried in an oven to get the saponin in the powderous form. Next, the Secondary sludge samples and inoculated cow dung were stabilized and stored at 4 to 5 degrees Celsius to arrest further degradation. Identical digesters, each with working volume of 130 ml, were used for control and experiment. The sludge and the inoculum were taken on the basis of F/m ratio of 0.5. F/m ratio is nothing but food to microorganisms ratio. All microorganisms were added to the digester at the concentration of 10 grams per kg of total solids, 20 grams per kg of total solids, and 30 grams per kg of total solids, which is nothing but 1%, 2%, and 3% concentrations. Next, digester setup. This is a model of a digester setup. It works on the principle of Mario's principle. You can see that there are two containers, one containing substrate plus inoculum, another water. The container which has water is fixed inverted to the clamp. The connections were made through the rubber tubes. The gas produced during digestion process is in the digester moves through the tube and collected in the container by displacing equal amount of water. The water displaced is collected in the vessel kept at the bottom. So the amount of gas produced was measured directly by measuring the amount of water displaced. We move on to the results and discussion. Microcalorimetric analysis. We did this microcalorimetric analysis as a preliminary step to check whether there is any effect on biomass when added with surfactants. So we took 1% supponent surfactant as test surfactant and carried out this analysis. This is a graph obtained from the microcalorimetry. Microcalorimetry works on the principle that any microbial activity accompanies with heat change. From the graph, it is understood that heat released by the sample with the supponent is nearly 2 times more than that by the sample with without surfactant. Keeping this as a basis, we proceeded our experiment. Next, effect of supponent on anaerobic co-digestion. As a next step, we did the digestion process for supponent at different concentrations. And this is a gas production graph. This graph shows that digester with 2% supponent has higher efficiency in enhancing the gas production. Though 1% supponent enhanced the digestion process, but not that much to 2% supponent. The digester with 3% supponent yields a gas less than that controlled digester. So we can say that 3% supponent addition inhibits the gas production. Next, effect of 1% surfactants on anaerobic co-digestion. As a next level, we perform digestion trying all these surfactants at 1% concentration. This is a graph obtained. This supponent has better efficiency in increasing the gas production and is nearly 2.6 times more than the control, whereas SLS comes next to supponent and it is 1.9 times more than control. The Triton is the least among the 3 surfactants in enhancing the gas production and is 0.6 times more than the control. Next, we tried for 4 surfactants at 2% concentration. This is a graph obtained. In this also, supponent has higher efficiency in enhancing the gas production and SLS comes next to supponent, which is 2.3 times more than the control. Triton has also enhanced the gas production but not as by the other 2 surfactants. At last, we perform the digestion process with surfactant at 3% concentration, and this is a graph obtained. In this, you could see that all the 3 surfactants inhibits the gas production. Supponent inhibits the gas production in lesser amounts and the gas production was 0.2 times lesser than the control, whereas SLS also inhibit the gas production and the gas produced by it was 0.4 times lesser than the control, and the Triton inhibits a gas production a lot when compared with other two surfactants at 3% concentration. This is a TS, and next we move on to TS and VS values. The TS and VS values were determined after the digestion process or presented here. In this, the volatile solids for the sample with 1% and 2% surfactants was decreased when compared to control, and the volatile solids values for the sample with 3% surfactants is higher than the control. So we could say that some volatile solids has been converted into gas. Next, we perform the modeling of gas production. Experimental gas production data obtained during the anaerobic digestion process was modeled using modified Gompertz model. This model suits best for all anaerobic digestion process. This is a expression for the model. In this expression, Y represents a gas accumulated, A represents a gas production potential, mu m represents a maximum gas production rate, and T naught represents a lag time of the process. Using the modified Gompertz model, we compared our experimental data with the theoretical values. So, this is a graph that is plotted for both experimental values and the theoretical values for the digestion process with 1% surfactants. In the graph, the symbol represents experimental values, whereas the trend represents the theoretical values. So from the graph, it can be seen that both experimental data and the theoretical data matches with each other very closely. And this is we'll move on to comparison for 2% surfactants using the modified Gompertz model. This is a graph for 2% surfactants. Experimental data and theoretical data matches closer to each other in this graph. Next, comparison for 3% surfactants. This is a graph for 3% surfactants. Here also, experimental data and theoretical data matches closer to each other, which shows that our experiment was moved in good progression. So, we conclude by saying that addition of surfactants at lower concentration leads to the enhanced gas production. This is the order of the surfactants according to their efficiency in enhancing the gas production. All the surfactants used improves the digestion effect at 2% concentration, whereas the digestion was inhibited at 3% concentration. Although the enhancement at lower concentration of surfactant can be due to increase in the bioavailability, biosorption, the significance of surfactant properties such as CMC, critical micellar concentration, molecular weight, hydrophobic chain length, iconicity, etc. could play an important role. The experimental gas production data obtained matches with the modified Gompertz model for all the concentration of surfactants. Thus, the gas production has also been verified theoretically. Thus, we end our presentation by saying that the surfactants which we used enhanced the gas production up to 2%, whereas it inhibits the digestion process at 3% concentration. So, thank you.